Hi everybody, and welcome to our module on innate immunity. The easiest way to understand the immune system at a basic level is to think of it as a series of barriers to infection. So let's imagine, for example, that we have a bacteria here, or maybe a helminth like I've drawn here, or maybe a virus like I've drawn here. And these pathogens want to get inside the body because inside the body they can grow and thrive. Well, the first barrier that keeps them from getting inside the body is the skin and the mucous membranes. We often don't think of this as part of the immune system, but the reality is that there are many pathogens we're exposed to every day that never gain entry to the body because they can't get past these basic layers. But once a pathogen gets past the skin or the mucous membranes, it encounters what most of us think of as the immune system. And the first thing it encounters when it gets past the skin and mucous membranes is the innate immune system. This is a system made up of cells like macrophages and neutrophils and proteins like complement, and they attack and try to kill these pathogens as soon as they get in the body. As we will learn, however, in this module and other modules, the innate immune system is not so good at dealing with overwhelming infections or dealing with viruses. So once a pathogen gets beyond the ability of the innate immune system to control the infection, it then will encounter the third barrier, and that is the adaptive immune system. The adaptive immune system is made up of B cells and T cells. These cells create an immune response that is highly specific to an invader. They devise systems to kill one specific invader. It takes time for them to mount their response, but once they do so, this is the most powerful portion of the immune system. Another feature of the adaptive immune system that's unique is that it has memory. So if you get infected by the same pathogen 2 times, the second infection will elicit a quicker and more powerful response from the adaptive immune system. So on this slide, I've summarized some of the key differences between the innate and the adaptive immune systems. The innate immune system is a fast-acting system. It is standing by, ready to attack any pathogen immediately if it gets beyond the skin and the mucous membranes. In contrast, the adaptive system is slow. It takes a few days to mount its response. But as I said before, once it does, it's a much more powerful response than what you get from the innate immune system. The innate immune system is a non-specific reaction. The same cells respond in the same way to many different invaders. The adaptive immune system is a highly specific response. As we will see in other modules, B and T cells proliferate in response to unique antigens on unique cells to try and repel and kill that invader. And then finally, the innate immune system has no memory, so a second infection elicits the same response as the first infection. The adaptive immune system has that important feature called memory, where the second infection elicits a faster and more robust response than the first. Before we can talk about the innate immune system, we have to define a number of terms that are very important for discussing immunology. And the first term is that of antigen presentation. So I want you to imagine that we have some tissue in the body here, and it has been infected by a bacterial cell. I'll draw a little circle around a B here for the bacterial cell. You should know that the innate immune system can react directly to antigens present on the bacterial cells. So for example, as we will see, macrophages can eat these bacteria. It's a process called phagocytosis, and they can do that directly just by sensing the presence of the bacteria. On the other hand, cells of the adaptive immune system, the most important of which are the T cells, cannot react directly to this bacteria. The bacteria can be right next to the T cell, and the T cell won't do anything at all. The only way the T cell can react to this bacteria is if the bacteria is first consumed by a cell called an antigen-presenting cell, or an APC. That antigen-presenting cell will then gobble up the bacteria and present little pieces of the bacterial peptides on its surface, and then the T cell can see the bacterial pathogen and react to it. So T cells in the adaptive immune system require antigen presentation in order to activate. And the reason this system evolved is because the T cell response is incredibly powerful. And if T cells could react to anything out in nature, they would probably react too much and we would die from autoimmune diseases. By requiring antigens to be presented to T cells by antigen-presenting cells, it fine-tunes the system so that the adaptive immune system only responds to important invaders. So I've summarized these points for you on this slide here. The innate system can be activated by free antigen, like that bacterial cell floating out in the tissue. Pathogenic molecules can be detected freely in the blood and tissue. On the other hand, the adaptive immune system, especially the T cells, require antigen presentation. Pathogens must be engulfed by cells and broken down. Pieces of the proteins have to be transported to the surface, and the antigen has to be presented to the T cells for activation. Another important concept for discussing immunology is the concept of cytokines. These are cell signaling proteins that are often released by immune cells, and they travel locally to stimulate an inflammatory response. And there are a number of subsets of cytokines, and you have to be familiar with these terms. So chemotaxis is the process of attracting immune system cells. So cytokines that attract immune system cells are called chemokines. Interleukins are another subset of cytokines. They were given this name because these proteins travel between leukocytes, and they're named with numbers like IL-1 and IL-2. Some cytokines are called tumor necrosis factors, and these were named because they can cause tumor cell death, but they also are used by white blood cells to trigger immune responses. And then other cytokines are called transforming growth factors and also interferons. The interferons got their name because they interfere with viral replication. And then the final concept we need to define before discussing the innate immune system is that of the CD structures, or the cluster of differentiation structures on cells. So CD structures are cellular surface molecules, and they have names like CD3 and CD4 and CD8, and they are found on many immune cells, especially T cells and B cells. And we use them to identify cell types. So for example, when we say we have a CD8 T cell, we're talking about a specific subtype of T cell, and it's defined by the presence of this CD8 molecule on its surface. Also, some of the CD molecules are used as receptors and cell binding sites, and we'll talk about those in the remainder of this module. So let's start our discussion of the innate immune system by listing the key players in this system. And the first set of key players are the phagocytes. These are cells that essentially eat pathogens that get inside the body. The first group are the macrophages. This is the hallmark cell of innate immunity, as we shall see. And then the second group are the neutrophils, which are cells that are essentially on call, and they can be called in to assist the macrophages when an infection is taking place. The next key element of innate immunity is the complement system. This is a series of proteins that can be activated inside the body to kill pathogens. And then finally, there are natural killer cells, eosinophils, and mast cells, and basophils. And in the remainder of this module, we're going to talk about all of these elements of innate immunity. So first, let's talk about how those elements of the innate immune system recognize pathogens that have gotten past the skin and mucous membranes. And you should know that all those elements of the innate immune system can recognize molecules that are foreign. And it does this by looking for structures on pathogens called pathogen-associated molecular patterns. These are patterns of molecules that are present on many microbes, but not usually present on human cells. And the way the innate immune system recognizes these structures is by the use of pattern recognition receptors. And one of the most important classes of pattern recognition receptors are the toll-like receptors, or TLRs. These are found on cells like macrophages and dendritic cells and mast cells, and we'll talk about this in a minute. And once these cells recognize the pattern-associated molecular patterns, or the PAMPs, they then secrete cytokines which trigger an inflammatory response. So what are some of the important pattern-associated molecular patterns that the innate immune system responds to? One of the most important ones is endotoxin, or lipopolysaccharides, which are found in gram-negative bacteria. Lipopolysaccharides can bind to lipopolysaccharide binding protein, which is found in plasma, and then that complex can bind CD14 on macrophages. And I've written this in bold because it's high yield to remember that CD14 binds lipopolysaccharide. Once CD14 is bound, it will trigger a toll-like receptor called TLR4, and this will lead to a sequence of events which leads to production of cytokines by macrophages, including IL-1, IL-6, IL-8, and tumor necrosis factor. We'll talk about all these cytokines in a few slides. The innate immune system can also recognize peptidoglycan in the cell wall of bacteria. If bacteria get inside a macrophage, for example, there are receptors called NOD receptors that are intracellular. NOD stands for nucleotide binding oligomerization domain, and that's a mouthful. But once peptidoglycan binds these NOD receptors, this leads to cytokine production. Other patterns that are recognized by the innate immune system include mannose. Mannose is a polysaccharide found on bacteria and yeast. And there is a protein in the body called mannose binding lectin that comes from the liver. And once it binds to mannose, this can activate the lectin pathway of the complement system, which we'll talk about in another module. Another pattern recognized by the innate immune system is lipoteichoic acid. These are proteins found on gram-positive bacteria. Also, double-stranded RNA. Double-stranded RNA is never found in normal human cells, but many viruses are RNA viruses. Some of them carry double-stranded RNA in their genome. Others have a single strand of RNA, but they will briefly become double-stranded when they are copying that RNA genome. So the innate immune system is programmed to recognize double-stranded RNA. Another pattern that the innate immune system recognizes is unmethylated DNA. So most of the DNA in human cells is methylated when the innate immune system sees lots of unmethylated DNA that is more commonly found in bacterial cells, and it will react to that. So now let's begin our discussion of the individual elements of the innate immune system, and we'll begin by talking about monocytes and macrophages. So macrophages are the guardians of innate immunity. You can just think of them as soldiers standing guard behind the skin and mucous membranes, ready to consume any pathogens that get inside the body. Monocytes are produced in the bone marrow and they circulate in the blood for about 3 days. You have billions of monocytes in your bloodstream all the time. When the monocytes enter tissues, they become macrophages, and they go by different names depending on which tissue they live in. When they live in the liver, they're called Kupffer cells. When they live in the central nervous system, they're called the microglia. When they live in bones, they're called osteoclasts, but all of these are macrophages performing the same function as macrophages anywhere in the body. So macrophages perform 3 vital functions for innate immunity. The first is phagocytosis, which we'll talk about in a minute. The second is to produce cytokines, which trigger the inflammatory response. And then the third function is to present antigens to the adaptive immune system. We won't talk about this much in this module, but just remember that the innate and adaptive immune systems are interlinked, and the cells of innate immunity, like macrophages, can present antigen to the cells of the adaptive system, like T cells, to trigger the adaptive system to respond. Phagocytosis is a process by which macrophages engulf pathogens and trap them inside a structure in the cell called a phagosome. This is a picture down here of phagocytosis, and there's a bacterium here, and it's being essentially eaten by the cell and then trapped inside a phagosome. The phagosome then merges with a lysosome, and the lysosome contains deadly enzymes. The molecules inside a lysosome are so deadly that if they were free inside the cell, they would kill the macrophage, but they're contained inside the lysosome to protect the cell. So once the lysosome merges with the phagosome, then you get death of bacteria or viruses or whatever pathogens have been engulfed by the macrophage. Let's talk about what you find inside those lysosomes that is so deadly to bacteria and other pathogens. Well, one of the first things you find are reactive oxygen species. These are called superoxides. These are molecules produced by an enzyme called NADPH oxidase as part of the biochemical pathway called the respiratory burst. The respiratory burst pathway leads to production of hydrogen peroxide and superoxide molecules, and both of these are toxic to bacteria and other pathogens. Also inside of the lysosomes, you will find reactive nitrogen intermediates. This happens when nitric oxide is reacted with those superoxide molecules. It creates compounds like peroxynitrate, and these are also toxic to bacteria and pathogens. And then finally, you will find many enzymes that break down structures in bacteria and other pathogens. These include proteases, which break down proteins, nucleases, which break down nucleic acids, and lysozymes, which hydrolyze peptidoglycan in the cell wall of bacteria. This is a picture of a lung abscess, and I've shown you this to remind you that the contents of the lysosomes, although designed to destroy pathogens, can sometimes be secreted and cause tissue damage. Macrophages and especially neutrophils in the setting of significant inflammation will sometimes release some of their lysosomal contents, and this is part of the way you get tissue destruction when you get lung abscesses. It's also part of the way you get tissue destruction in many autoimmune diseases. For step 1 of your boards, it's important that you know the clinical correlates of this process of phagocytosis. And the first clinical correlate is that some pathogens block the process of fusion of the phagosome with the lysosome. And the most important one is tuberculosis. It modifies the surface of the phagosome so that it can't fuse with the lysosome. And this is what allows tuberculosis to proliferate inside of macrophages. This also helps it to evade antibodies which are in the plasma and tissue and can't get inside the macrophage. There is also a rare immune deficiency syndrome called Shediac-Higashi syndrome, which results in failure of lysosomes to fuse with phagosomes, and children who are born with this rare immune deficiency have recurrent bacterial infections. Macrophages can exist in several states. In the resting state, the function they perform is debris removal. They circulate through tissues and pick up dead and dying cellular tissue from the human body. In the activated or prime state, they become more effective phagocytes and killers. And the major activators are those pathogen-associated molecular patterns we talked about before that use toll-like receptors to activate the macrophage. This includes things like lipopolysaccharides from gram-negative bacteria, peptidoglycan from bacterial cell wall, and bacterial DNA, which is relatively unique because it has much less methylation than human DNA. Other things that can activate macrophages include interferon gamma from T cells. So when the adaptive immune system and the T cells become activated, They can then in turn stimulate the macrophages to become more activated. Also, natural killer cells secrete interferon gamma. And then finally, when the complement system is activated, it produces a protein called C5a, which we'll talk about in the module on complement, and C5a can attract macrophages to the site of bacterial pathogens and other invaders. On this slide, I've listed some of the important surface receptors found on macrophage cells. CD14 is the surface receptor that binds to lipopolysaccharide in gram-negative bacteria. There is an Fc receptor that can bind to antibodies. It binds to the Fc portion of antibodies. We'll talk more about antibodies in the module on B cells. There is a receptor for C3b, which is a complement protein, and this allows macrophages to bind anywhere that complement is found. And then finally, there are three molecules that allow macrophages to interact with T cells. First, there's MHC class II, which binds to CD4 on T cells. B7 binds to CD28 on T cells, and CD40 binds to the CD40 ligand, and I talk about all of these structures in the module on T cells. Macrophages release several cytokines that you need to know, especially for step 1, and the two most important ones are IL-1 and tumor necrosis factor alpha. Some others that we'll talk about are IL-6, IL-8, and IL-12. IL-1 and tumor necrosis factor both increase synthesis of endothelial adhesion molecules, and this allows neutrophils to enter inflamed tissue. We'll talk more about this in a minute when we discuss neutrophils. Macrophages secrete IL-1, and I've always found this easy to remember because macrophages are the first line of defense in innate immunity, and therefore it makes sense that they secrete the first interleukin. IL-1 is an endogenous pyrogen. A pyrogen is anything that causes fever, and IL-1 is endogenous because it comes from within the human body. An example of an exogenous pyrogen would be lipopolysaccharide, which comes from bacteria. IL-1 acts on the hypothalamus and it raises the set point for temperature in the body. Tumor necrosis factor alpha can cause vascular leak. It's one of the reasons people can develop septic shock when they have an overwhelming infection. TNF alpha is called a cachectin because it inhibits lipoprotein lipase in fatty tissue. The result is there's reduced utilization of fatty acids, and this is why people can become cachectic when they have an infection. Tumor necrosis factor alpha has been shown to kill tumors in animals. That's where it gets its name, tumor necrosis factor. And the way it kills tumors in animals is by causing coagulation and thrombosis in the blood vessels that supply the tumor. Therefore, the tumor dies. And this can help you remember that it can also cause intravascular coagulation and DIC in patients who have severe infections. Other cytokines produced by macrophages include IL-6, IL-8, and IL-12. IL-6 is also a pyrogen like IL-1. It causes fever. IL-6 also stimulates the liver to synthesize acute phase proteins like C-reactive protein, which become elevated when there is inflammation in the body. IL-8 attracts neutrophils, and IL-12 is important for promoting the development of the Th1 subset of T cells, which is part of the cell-mediated response, and I talk in more detail about this in the module on T cells. The next element of the innate immune system is the neutrophil, and the easiest way to think about neutrophils is that they are the backup cells to macrophages. When macrophages encounter pathogens, they can call in neutrophils from the bloodstream for help. Neutrophils are derived from the bone marrow, and they stain pink if you stain them with right stain. This is a picture of some neutrophils down here in the bottom right-hand corner of the screen. By comparison, eosinophils, which we'll talk about in a little bit, stain red, and basophils stain blue. Neutrophils circulate in the bloodstream about 5 days, and they die unless they're activated. They are drawn from the bloodstream to sites of inflammation by macrophages and cytokines. We'll talk about that process in a minute. Once they enter tissues, their job is to do phagocytosis. The granules that stain pink with right stain are lysosomes, and they contain bactericidal enzymes and all those things we talked about before that can kill bacteria. And as I said before, just think of these cells as providing backup support for macrophages when there's an infection taking place. The process by which neutrophils exit the bloodstream to enter sites of inflammation is very high yield to know for your boards, and it's been worked out in detail over the last several decades. So neutrophils that are circulating in the blood first start to slow down when they get near sites of inflammation, and this process is called rolling. They slow down and roll along the blood vessel. The reason they begin to roll is because ligands called selectin ligands on neutrophils bind to the endothelium. And these ligands contain a carbohydrate called sialyl Lewis X. This is a very important and famous carbohydrate structure. It's the way the neutrophils bind to the endothelium. It's also the way that sperm cells bind to eggs for fertilization. These ligands on the neutrophils bind to molecules called E-selectin or P-selectin on endothelial cells, and this makes them slow down and start to roll when they are traveling through the bloodstream. The next thing that happens is they begin to crawl. This is a process called tight binding, and this occurs because neutrophils express a molecule called integrin on their surface, and integrin binds to intercellular adhesion molecules, or ICAM molecules, on the surface of endothelial cells. After they're crawling, they begin the process of transmigration across the endothelium and into tissues. This occurs because neutrophils bind molecules called PCAM1, which are found between endothelial cells, and then they migrate to the site of inflammation drawn by chemokines like C5a from the complement system or IL-8 from macrophages. So here are some pictures to illustrate this process and to help you remember it. So neutrophils are always circulating with selectin ligand on their surface, but normally there's nothing for it to bind to, so they continue to circulate through the bloodstream. However, when infection and inflammation occur, macrophages release IL-1 and tumor necrosis factor, and this stimulates the expression of selectin on the surface of endothelial cells like I've drawn here. As a result, the neutrophils will bind to the selectin and they will begin to roll along the endothelium. For crawling to occur, the neutrophil needs to express integrin like I've shown here, and the triggers for integrin expression are other components of infection and inflammation like lipopolysaccharide or C5a, the things that stimulate neutrophils. They put integrin on their surface and then they can bind to ICAM. ICAM is always present in endothelial cells, but normally it has no partner to bind with on the neutrophils, but in settings of infection and inflammation, these types of things stimulate the expression of integrin, and then the neutrophil can bind to ICAM, and that's how it begins to crawl. Neutrophils contain two groups of granules, and you should know something about the differences. The first group are the small granules. These are also called specific or secondary granules. They contain a number of enzymes. I've listed some of the important ones here. Alkaline phosphatase is an enzyme from the small granules, and you may know that in certain types of leukemias, patients have what's called a low alkaline phosphatase score. This means that they have neutrophils in their bloodstream, but they have very low levels of alkaline phosphatase, and this is seen in chronic myelogenous leukemia. The small granules also contain collagenase, which breaks down collagen, lysozymes, which break down the peptidoglycan cell wall, and they also contain lactoferrin. Lactoferrin gets its name because it's found in breast milk and it binds iron, but in terms of the neutrophils, what it can do is lyse the cell and kill bacteria. These small granules fuse with phagosomes and they kill pathogens. They also can be released into the extracellular space. The larger granules are called azurophilic or primary. These contain enzymes like acid phosphatase, which is an enzyme that can degrade proteins in an acidic environment, so they work very well in the acidic granules, and also myeloperoxidase, which is an important enzyme for the respiratory burst that kills bacteria. And these larger granules only fuse with phagosomes. They aren't released into the extracellular space. And finally, you should be familiar with the neutrophil concept of band forms. This is a picture of some band forms. These are neutrophils with a band-like nucleus, and these are immature neutrophils. And they're seen in the plasma during bacterial infections when the bone marrow is being driven to produce more neutrophils. And clinically, people often describe this as a left shift. The reason they call it a left shift is historical. There used to be a keypad for counting cells when you looked at a blood smear, and it had a key for red cells and white cells and platelets. And over here on the left side of the keypad was the key for the band forms. So when you saw lots of bands, you would click this left side of the keypad a lot, and so therefore it was called a left shift. And people still use this term to indicate that there are band forms present in the bloodstream, and this means that there's evidence of infection and inflammation. It's important to remember that neutrophils do not present antigen. They perform phagocytosis only, and this is in contrast to macrophages, which are both antigen-presenting cells and also phagocytes. There are a couple of well-described chemotaxins. A chemotaxin is an attractor of another cell. There are a couple well-described chemotaxins for neutrophils. Those include IL-8, which comes from macrophages, and also C5a, which comes from the complement system. And then finally, opsonins are substances that bind to a foreign invader and increase the likelihood that it will be taken up by phagocytosis. And a major opsonin for neutrophils is IgG. Neutrophils have receptors for the Fc portion of the IgG antibody, and this is the only antibody that significantly binds to neutrophils to enhance phagocytosis. We have an entire module on the complement system, but for our purposes here, I just want to mention that complement is a very important component of the innate immune system. Complement proteins are produced by the liver, and the most abundant and important complement protein is C3. It's central to all the pathways of complement activation. And there is frequent spontaneous conversion of C3 to C3b via hydrolysis. This is called the alternative pathway of activating the complement system. Now, if C3b doesn't find something to bind to, it's rapidly destroyed. However, it can bind to amino and hydroxyl groups, which are commonly found on the surface of pathogens. This is an amino group here and a hydroxyl group here. So if C3b finds one of these, it can bind and become stabilized. Once C3b becomes stabilized, it leads to formation of the MAC, which stands for the membrane attack complex. And the process of this is drawn down here on the bottom of the screen. And the MAC ultimately forms pores in bacteria, leading to cell death. And we'll talk about this in more detail in the module on complement, but just remember that this is an important part of the innate immune system. The next element of the innate immune system are the natural killer cells. These are cells that have two very important roles in innate immunity. First, they kill human cells that are infected by viruses, and secondly, they produce interferon gamma, which activates macrophages to get them into that primed or activated state. In the module on T cells, I'll talk at length about MHC class 1, but for our purposes here, just know that it's a surface molecule found on most human cells, and it presents antigens to CD8 T cells to activate the adaptive immune system against intracellular pathogens, especially viruses. Unfortunately, some viruses have evolved mechanisms to block MHC from getting to the surface of cells. So one function of natural killer cells is to destroy any human cell that does not have MHC class 1. If MHC class 1 is missing, the natural killer cell thinks it must be infected with a virus and it destroys that cell. And this is a very important way we protect ourselves against some viruses that can block class 1 expression. There are two important surface molecules on the surface of natural killer cells. The first one is called CD16. This binds to the Fc portion of IgG antibodies. We'll talk more about IgG antibodies in the module on B cells, but for now, just know that it has an Fc portion to the antibody, and this can bind CD16 and natural killer cells and lead to enhanced activity of the NK cells. It is through CD16 binding of the Fc portion of IgG that natural killer cells perform antibody-dependent cell-mediated cytotoxicity, and I'll talk about this in a minute. The other surface molecule that's important to know about is CD56. This is also called NCAM, which stands for neural cell adhesion molecule. It's a commonly used marker to identify natural killer cells. When they find that cells express CD56, they know that they are natural killer cells. It's also found in brain and neuromuscular junction, and it aids to binding of other cells, but its function is not as important as knowing that it's a marker for natural killer cells. Let's talk about the process of antibody-dependent cellular toxicity. This is a process by which the adaptive and innate immune systems work together to kill a pathogen. In this process, antibodies, which are produced by B cells of the adaptive immune system, coat a pathogen or cell, and this is illustrated in this picture down here. IgG antibodies are coating this cell right here. The pathogen is then destroyed by immune cells in a non-phagocytotic process. And the classic example of the cells which kill the pathogen are natural killer cells or eosinophils. So importantly, these are elements of the innate immune system working together with antibodies, which are elements of the adaptive immune system. In this picture here, the CD16 molecules of the NK cells bind to the Fc portion of the IgG antibodies, and then the natural killer cell degranulates, and this causes death of the cell. The two classic examples of antibody-dependent cellular cytotoxicity are natural killer cells and eosinophils. In the case of natural killer cells, like I showed you before, IgG binds to pathogen-infected cells. The CD16 molecules on the natural killer cells bind to the Fc portion of IgG, and then the natural killer cell kills the pathogen. In eosinophils, the important antibody is IgE. It binds to pathogens, especially large parasites, and then eosinophils bind the Fc portion of the IgE antibody. Then they release toxic enzymes onto the parasite. Some more important features of natural killer cells. They are lymphocytes, so they are the same lineage as T cells and B cells. However, natural killer cells are members of the innate immune system, whereas T and B cells are members of adaptive immunity. They do not mature in the thymus like T cells, they have no memory like T and B cells, and they do not require antigen presentation by MHC as T cells do. So they are often identified as very unusual members of the lymphocyte group because they're very different from the T and B cells. The last elements of innate immunity are the eosinophils, the mast cells, and basophils, and I find it helpful to think of these three groups together. They all contain granules with destructive enzymes. They can all be activated or triggered by IgE antibodies from B cells. They're all important for defense against parasites, especially helminths. These pathogens are way too big to be phagocytosed by macrophages, so these cells probably evolved in order to protect us from these large invaders. What they do is release toxic substances onto the parasite to kill it without having to engulf it in phagocytosis. And unfortunately, their main medical relevance is in allergic disease. Although they're intended to kill parasites, we don't often encounter many parasites these days, but many people have allergic reactions, and these are triggered by these cells. Let's talk about the eosinophils first. Eosinophils have granules that appear red when you stain them with right stain. This is a picture of an eosinophil on the bottom right side of the screen. The reason they stain red is because one of the components of their granules is a substance called major basic protein. It has a positive charge, and it picks up the negatively charged eosin dye, which is a component of the right stain. What eosinophils are designed to do is discharge their contents onto parasites. Remember, they're designed to fight parasites like helminths that are too big to be phagocytosed. So their granules contain cytotoxic enzymes that they're supposed to dump onto the parasites, and these include substances like major basic protein, eosinophilic cationic protein, eosinophilic peroxidase, and eosinophilic-derived neurotoxin. And you can read a lot about these. We know a lot about many of them because eosinophils are important in allergic disease, so we've studied these greatly to try and understand how to prevent allergies. Eosinophils are activated by the IgE antibody, and I talked about this a few slides ago when I described the process of antibody-dependent cellular cytotoxicity. Eosinophils are stimulated by IL-5, which can be released by T cells. The Th2 subset of T cells releases IL-5, and I talk about this in more detail in the module on T cells. And you should know that high eosinophil counts are seen in patients who have a helminth infection. Normally, eosinophils are less than 5% of your white cells, or less than 500 eosinophils per microliter. The percentage of white cells goes up when patients are infected by a worm or a helminth. You will also see elevated levels of eosinophils in many allergic diseases. Mast cells and basophils I like to think about together because these two cell types share many similarities. First of all, they both contain granules that appear blue when stained with right stain. This is a picture of a basophil in the bloodstream shown here on the screen. The two cells are different in that basophils are found in the bloodstream and mast cells are found in tissue, but both cell types can bind the Fc portion of IgE antibodies, and when those IgE molecules cross-link on the surface of the cells, this leads to degranulation. This is easier to understand with a picture, so if you have a mast cell here, what it will do is bind lots of IgE antibodies so that they are found on the surface like this. The Fc portion is right here bound to the cell, and the antigen binding portion of the antibody is sticking out. When a pathogen comes along, it will cross-link multiple of these IgE antibodies together, and that cross-linking will trigger the cell to spill its contents. And the contents of the cell that get spilled when the IgE molecules are cross-linked include, very importantly, histamine, which is a molecule that leads to vasodilation, also many enzymes like peroxidases and hydrolases that are designed to kill parasites in the tissues. So we have now talked about all of these components of the innate immune system, and I'm going to talk about these components of the adaptive immune system in other modules. There are the T cells, which include the CD4 subset, which produces cytokines, and the CD8 subset, which is important for destruction of infected human cells. We will also talk about B cells in another module, and B cells are what produce antibodies. But for our purposes here, just remember that the adaptive and innate systems are interrelated. So the cytokines from the innate system activate these cells in the adaptive system and vice versa. And then the innate immune system, especially macrophages, will present antigen to T cells to activate the adaptive immune system. Finally, I want to mention a very important cell type, and that's called a dendritic cell. It's also called a Langerhans cell when it's found in the skin. And these are star-shaped cells like in this drawing here. They also look like this electron micrograph right here. And they're found in the skin and mucosal membrane. And what these cells do is they are professional antigen presenters. So they pick up a sampling of the antigens found in the skin and mucosal membrane, and they migrate to the lymph nodes where they activate T cells. You can think of these cells as a go-between between the innate and adaptive immune systems. So if you have some tissue out here in the periphery and there's a battle raging with macrophages and neutrophils fighting a pathogen, what these dendritic cells do is take a sampling of the antigens and go to the lymph nodes, which is where you find the T cells. They will then stimulate the T cells, which can then come back and help to fight the infection out here in the periphery. As I said before, they're like a communicator or go-between for the innate and adaptive immune systems. This slide is to familiarize you with some of the terminology used when describing the cells of the immune system. So granulocytes are cells which contain granules. So those include the mast cells, the eosinophils, the basophils, and the neutrophils. Of these four, the neutrophils are far and away the most predominant type. So often when people say there's a low granulocyte count, they really mean there's a low neutrophil count. And then agranulocytes are white blood cells that do not have granules. So those include the monocytes, which form macrophages in the tissues, and then the lymphocytes, which form the B cells and the T cells and the natural killer cells. And just remember, the natural killer cells are very unusual in that they are lymphocytes, but they are members of the innate immune system. Finally, let me describe the lineage of immune cells, or the way they develop from stem cells in the bone marrow. So a multipotential stem cell can differentiate into a lymphoid progenitor or a myeloid progenitor. The lymphoid progenitors can become natural killer cells or can become T cells or B cells. The myeloid progenitor goes on to develop megakaryocytes, which become the platelets or red blood cells, and they can also form myeloblasts, and myeloblasts can differentiate into basophils, neutrophils, eosinophils, or the monocytes, which then go to the tissue and form the macrophages. And that concludes our module on the innate immune system.